All right, welcome everyone. We're very excited to moderate this session entitled Diet and the Gut Microbiome. My name's Aaron Hecht. I'm one of the co-moderators of this session.
And I'm Christina Martinez-Kern.
We're really excited to have a terrific lineup of speakers today. We have 6 abstracts that were selected as some of our very best in this space, and we're very excited to hear them speak today. Our first speaker is Fred Warren from Quadram Institute Bioscience, who will be telling us about spatial temporal profiling of the human small intestinal microbiome.
Great. Thank you for the introduction and for the opportunity to talk to you today. And well done, everyone, for sticking it out to the last session of DDW. So I'm going to talk to you today about some of the work that my group have been doing at the Quadram Institute in Norwich in the UK, looking at profiling of the impact of diet on the small intestinal microbiome. So I think we've seen this figure quite a few times through the conference. I'm sure I don't need to tell this audience that the gut microbiome's very important for a whole range of different regions. Most studies that look at the gut microbiome focus on the colon. It's the most densely populated microbial community, and it's also the easiest one to access because we can take stool samples. However, if we look through the whole intestinal tract, there are a range of different environments with different chemical and physical parameters which give rise to different niches. So moving from the stomach, which is very acidic, the pH increases as you go through the small intestinal tract, the oxygen levels go down and the mucus layer changes, and all of this can impact the microbiome that we find in different regions of the small intestine. So studies that have looked at the small intestine microbiome are fairly few and far between because of the difficulties of accessing the small intestine. But they have identified that there's essentially a core intestinal small intestinal microbiota. And then there are segment-specific microbiotas in different parts of the small intestine, moving from the duodenum, which is a microbial community that really kind of resembles the oral cavity, a lot of species which are kind of shared with the oral cavity, through to the ileum, which begins to resemble more the colon. Most microbiome studies of the small intestine have either relied on biopsy or have relied on capsule sequencing, and we've seen quite a few studies looking at capsules which capture a part of the small intestinal microbiome throughout the DDW this week. And they're really great for getting a snapshot of what's going on in the small intestine, but they have limitations. So with biopsy or with capsule, you have to give them in the fasted state. You can't have any food with them. They only give you a snapshot of one small area, and with the capsules it may be difficult to work out where the sample's coming from. You know it's from somewhere in the small intestine, but it's a little bit difficult to say where. So what we wanted to do in this study was to understand not what's happening to the microbiome in the fasted state, but what's happening to the microbiome in the fed state. Of course, the fundamental function of the GI tract is food. We eat quite regularly. Food goes through our GI tract, and it potentially has a big impact on the environment throughout the small intestine. It provides nutrients. It also provides physical movement through the GI tract, and we wanted to explore that. So in order to do that, we were very lucky to work with the team of Gary Frost at Imperial College in London. Gary has a very unique clinical setup. And this is a photo of Gary in his clinical setup. So what he does is he has specially adapted nasogastric, nasoduodenal, and nasoileal tubes, which can be placed in volunteers, and then samples can be aspirated from the lumen of the small intestine, allowing us to directly access samples from the small intestine. The volunteers that do this are amazing. They have the tubes put in on the first day. They then live in the facility for 5 days, and they get fed a different meal on every day. And we can take time courses of sampling through having a meal, so we can follow that meal all the way down through the GI tract. And that's what we've done in this study. So we wanted to look at the impact of food structure. So we've read 3 different chickpea meals either clusters of chickpea cells, so kind of like a hummus, isolated chickpea cells, so a lesser level of structure, or broken chickpea cells, so basically gram flour. So 3 different levels of structuration in the meal. In this talk, I'm going to focus on the ileal samples, which were sampled every 30 minutes for 480 minutes postprandially. And we did both metabolome and microbiome analysis. I'm going to focus on the microbiome. We did shotgun metagenomics and metagenome assemblies from the shotgun metagenomics. So our results, the first question is, does the microbiome change when we eat a meal in the small intestine? And the answer is yes, it does. So this is just looking at Shannon index over time for our 3 different meal types. And you can see here the time 0 is the fastest microbiome, and then a meal is given. That meal gets into the ileum about kind of 60 to 120 minutes postprandially, and you can see there's a big increase in microbial diversity, particularly with the first 2 meals, but all 3 meals you can see there's an increase in microbial diversity. So when the bolus is passing through the ileum, it is having a direct impact on the composition of the microbial community. And we can zoom in and look at that at species level and see what shifts. So at baseline, we see a community that's really dominated by Haemophilus and Streptococcus, and this is consistent with what those capsule sampling studies say. However, you can see that there's distinct shifts over time. So this is for each of our 3 meals, and this is a time series in this heat map. So coming from baseline and then through a time series. And you can see at that kind of 120-minute or so time when our meal is getting there, there's this distinct shift in the microbial community. The participants were also given a second standard lunch, and you can see there's a second shift when they have the lunch. And we get a lot of species, things like Rophia, Eubacterium, Streptococcus salivarius appearing with the meal. And we thought to ourselves, well, that's kind of interesting because those really sound like oral species that are suddenly appearing in our sample. So we thought, well, maybe we can work out, are these actually oral bacteria that are being passaged in there? So we went back to our participants, asked them for one more thing, and got a saliva sample for them and sequenced that. And we then did strain sharing analysis. Could we have MAGs? We could do strain sharing, and we could identify if strains were shared between the oral cavity and the ileum. And we identified 1,500 or so strains in total. Of those, 100 were shared between the oral cavity and the ileum. And what we then looked was the proportional abundance of the strains that were shared between oral cavity and ileum. And what we found was that in the fasted state, about 10% of the strains were shared. But over time that increases and it goes up as high as 50% when the bolus passes through. So you seem to get a really big increase in oral strains appearing in the ileum over time. We can then look at the impact of that on the network structure and the network stability in the ileum. At baseline, what we can see is we've got these 2 quite tight clusters in the co-occurrence network of species. So we have one cluster, and these are colored by degree of strain sharing. So blue is things oral, red is things not oral. You can see this tight cluster of red species here. These are all kind of gut anaerobes. I know you can't really read the names, but they're things like F, Proubacteroides, etc. And then on the right-hand side, you have more oral species. What happens when the meal passes through is that network structure gets disrupted. So this is typical of a much more dynamic, unstable microbial community. You can see that the gut species have pretty much disappeared, the colonic anaerobes. And really the main species in the network are the oral species in blue. We can also think what this is doing from a functional level. So this is looking at KEGG pathways. And you can see, again, there's a shift in KEGG pathways, as we saw with the taxonomic data. And there's a move towards more saccharolytic pathways, so sugar metabolism pathways, also lactate and formate metabolism. And we can then ask what that's doing to the metabolites. So this is looking at individual interactions from those network graphs between metabolites and individual bacterial species. At baseline, we don't really see anything, and we kind of interpret that as the bacteria acting as a whole community. So there's not one-to-one relationships between bacteria and metabolites, but then when the food bolus appears, we start to get interactions between particularly those oral species and metabolites. particularly formate, some of the bile acids, and also acetate and short-chain fatty acids as well, and some amino acids. And what we're then interested to pick apart is what the impact of this is on gut hormone signaling. So we can, from our data, we have ileal GLP-1 and PYY levels, and you can see that there's an initial peak in both of these hormones after meal ingestion, and then there's a second phase of what we're calling extension phase. So we were interested to see if the microbes might be associated with this. So we looked at the association between microbe and hormone levels, and we found some associations between a number of microbes, particularly the oral microbes with PYY levels. And we then tried to pull apart what metabolites this might be linked to. Our current hypothesis is that formate may be very interesting. So what we find if we look at the short-chain fatty acids on this plot, and again, this has changed over time, a baseline, you get a lot of acetate propionate and butyrate because you've got all those colonic anaerobes in there fermenting away in the fasted state. That kind of disappears, it gets washed out by the bolus. You then get an increase in formate. Presumably those oral bugs are coming in, they're saccharolytic, they produce formate and lactate. So you get a sharp increase in lactate, and that is sort of correlated with that long-term, with that potentiation of gut hormone signaling. Obviously, we still need to do a lot of work to kind of pick that apart. So in conclusion, we've probably for the first time, we think, characterized the ileal microbiome and how it changes during a meal. We found that it's really dynamic. It changes on a minute-by-minute basis. And we go from a colonic anaerobe-dominated environment to an oral microbe-dominated environment. And by strain level analysis, we can show that these are actually oral bugs that are turning up there, not just the same taxonomy. The oral microbes in the ileum do modulate the metabolic environment. We see a load of different metabolites which can only be coming from bacteria, and that potentially has an influence on the release of gut hormone and signaling in the ileum. So finally, acknowledgements. So huge thanks to Gary Frost's team. None of this would happen without them, particularly Ming Zhu, who ran the human study and helped put the tubes down people. Nadia did a lot of the metabolite analysis, and Falk Hildebrand's group, who did the metagenomics and strain sharing, particularly the strain sharing analysis in the metagenomics. And thank you all for listening, and I'm happy to take questions.
This presentation's open for a couple of questions.
Hi, very good talk. I'm Rishan from King's College London. I have a question about, like, for example, now people take oral GLP-1, and what do you think the dynamic microbiome change has any guidance on that?
That's a super question. I have to admit that's not something I thought about. I don't know if it would have an impact. It might do. I guess it could have an impact on uptake of the GLP-1. It might have an impact on, it might be something you want to consider whether you're taking that with or without a meal, because that's going to have quite a big impact on the ileal environment when that oral GLP-1's getting there. So yeah, it's definitely something to consider. Good thought.
That was interesting. Just maybe a simple question. Did you determine the bacteria that were in the food? And did that play any role in the increase in diversity that you saw?
So that's a good question. We're doing those experiments at the moment. So what we're doing at the moment is we're taking some of the food preparations, sequencing them, getting people to chew and spit, sequencing again to see what comes out. Mm-hmm. We don't see anything in the bugs that aren't kind of normal human commensals, so I wouldn't suspect it's something. We had another study looking at duodenal microbiome where we did see Bacillus cereus with one of the foods, so we thought kind of maybe actually should we have been feeding that? Maybe there was a bit of contamination on one of the foods we were using, but in this study we didn't see anything that you would say would be a food microbiome associated bugs. They were all human microbiome associated bugs. So kind of indirectly we can rule that out, but we're going to go and do the experiments to go and check that out.
Great, thanks.
All right, thank you very much.
Thank you.
All right, next up we have Yael Haberman, and the talk title, or abstract title I should say, is Can Gut Microbial Indices Supplement Routine Health Checkups Using Gut Indices as Indicators for Consumption of Processed Foods, Adherence to Mediterranean Diet, Cardiac Stress, Ergometry, and Well-Being.
Thank you. Thanks. Thanks the organizers for the opportunity to present. I'm a physician scientist and I spend my academic effort between Cincinnati Children's and the Sheba Medical Center. And in the last 10 or so years, we have invested most of our effort in characterizing host microbial interaction and particularly using diverse and very deeply characterized human cohorts. I have no conflicts of interest. And really the motivation for doing this is, I think we heard this quite a bit during this meeting, is the increases in many diseases like obesity, IBD, celiac, EOE, colon cancer in the young. And the phase that this happened cannot be explained by genetic changes because genetic doesn't change so rapidly, but rather by dietary, environmental, and lifestyle changes. In addition, we see this heterogeneity that we see between different people across the globe, but also within the same family. I have siblings with Crohn's disease that I take care of, and each one of them, even in the same family, react different to the treatment that I give them. And again, this heterogeneity cannot be explained just by their genetics because we share many of our genes. And instead, we probably share like a smaller fraction of our gut microbiome. And really, it's a big question, how much of the exposure, lifestyle and diet do we share with each other? So again, the motivation in characterizing this heterogeneous large cohorts is to really to try to understand how we can maybe affect, redirect, prevent those diseases. And again, the motivation is to study this in human cohorts because it's very, very difficult to recapitulate diet, bugs, and exposures as humans in animal models. So this is a paper that we recently submitted and published. And here we showed how transition from a more rural to urban environment in China. We had the nice collaboration between the different sites, including a site in China. So how this transition from a more rural to urban area can be reflected in the feces. And we showed that time spent by rural residents in urban environment is really linked to changes both in the gut microbiome and the metabolomics, which kind of mirrored some of the changes we saw in the same ethnic population also in Crohn's disease. And then we wanted to look into this and a little bit ask more of a philosophical question. So how are we defining health in general? And more specifically, more practically, how do we define a healthy microbiome? And, we keep saying dysbiosis, but, it's not something that is really descriptive. So the WHO defined health as a state of physical, mental, social, and well-being, and not just merely the absence of disease. And then we asked, can the microbiome, which is part of our human ecosystem, which is exposed to the same exposure, same diet as we are, can be used, like we saw in the rural-urban transition, as a more general tool to assess health across these different diverse dimensions. And to do this, we initially did this nice meta-analysis that was also published a couple of years ago. And here, instead of looking at comparison between one disease and a control group, we decided to compare across different disease cohorts. And we really downloaded from the public domain different disease cohorts that really encompass different aspects of health. So we had inflammatory bowel disease, we have some infectious disease, psychiatric disease, and neurological disease. And what this helped us is really to identify a shared signal that is changed across across most of these different diseases. And it helped us to build a tool that is composed of bacteria that are found in different controls, divided by the ones that are higher in disease, and we log transform this, and now we use a tool which is a very robust tool, which we call Microbial Health Index, and we really applied this after this. We generated this and validated this. We applied this to many of our new cohorts, and it really robustly held up very nicely to separate cases from controls. So now we move on to our new initiative, and this initiatives include 712 individuals with a mean age of 57, and of them were 40% females. And those people are yearly coming to an annual checkup as part of what is called as an executive medical checkup at the Sheba Medical Center. And the nice thing is that we have a very large and very deeply characterized cohort. We have access to the medical record, and there we have ICD-10 disease codes, we have the ATC medication classes, they get checkups, so we get current lab, CBC, lipid profile, and hemoglobin A1C, they get stress test, and they have a screening test even for glaucoma. And then we added to this a very thorough lifestyle questionnaire that includes physical activity, sleep habits, cooking, well-being, bowel movements, and alcohol and coffee habits. We included also a validated computerized FFQ, which we got very, very detailed information about dietary exposure. And we also had biospecimens. So we are looking at each of those components by itself, but also for simplicity, to understand what's going on here, we also came up with a medical record index with a lifetime index. We classified NOVA for ultra-processed ratio of exposure in each of this individual. We classify also adherence to the Mediterranean diet. And again, we use our Microbial Health Index and the FAIF Index to try to understand what's going on here within the different components. We try to keep most of our index in the same direction. So higher index indicating better health. So let's keep this in mind. So here we just look at some anecdotes of this cohort because I think it really reflects what we are going to see. So for example, people at a higher age acquire more diseases, and this is something that we can see at the EMR index. However, despite that, they practice a much healthier lifestyle. They adhere more to the Mediterranean diet, they practice a better lifestyle index, and they're exposed less to ultra-processed food. We didn't see many differences between females and males. We just saw that the males are exposed to more ultra-processed food in comparison to women, which was quite amusing. And then we did different ways we wanted to QC our data, and this is another nice anecdote coming from this data set. So we looked at how much the vitamins are coming from supplements versus diet, and how much is reflected at the end of the day when we measure the vitamin levels in the blood. So we did this for folic acid, and for folic acid, the main source that reflected the level in the blood was the amount coming from the diet itself. For B12, it's kind of separated between the diet and the supplements, and for vitamin D, it mainly came from the supplement. It makes a lot of sense because, again, to get the vitamin D, we don't just need the diet, we need the sun exposure to get it to the right levels. So this is just the metadata itself. Now we are moving to connect this to the microbial indexes. So here we saw a very nice correlation between the lifestyle index and our health index and alpha diversity. Similarly, we saw that people who are exercising more significantly had a better health index and some correlations with faith alpha diversity. Sorry, this was the well-being. So for well-being, we asked them how satisfied they are from their general condition. And for physical activity, we asked them of how many times per week they're exercising more than 15 minutes. And once again, we saw the negative correlation with the health index and with the alpha diversity also linked with the processed food. So here we are just showing all the components by itself and all the indexes. So green means positive correlation, usually again trending to better lifestyles or better indexes. Red shows the opposite way. If it marks with a plus, it means that it passed the FDR-Q correction. And the nice thing here that we are trying to show and reflect is that those correlations holds up when we look at all the cohort, when we looked at males separately, females separately, young and old separately, and also in people that didn't have any EMR-defined diseases. So some highlights. So we saw that higher fibers, higher nuts, adherence to the Mediterranean diet and vitamin C is linked with better health index and to some degree alpha diversity. Red processed food, ingestion, salty snacks, sugary drinks, adherence to processed food was linked to decreased indexes. And what was also interesting that this correlation is also persistent also when we looked at people who had hypertension, prediabetes, or taking lipid modifying agents, telling us that even within a more diseased population, a healthier lifestyle matters. Here we look at the lifestyle indexes, the labs, the medications, and the diseases. This is just highlights. So Baumwoman showed some interesting correlations. So positive correlation with health index, but negative correlation with faith diversity, which has been probably previously published. So again, indicating the validity of the data sets. The MET score, which is an outcome from the stress test that tells us something about fitness, was also very robustly correlated with the better indexes. The lipid profiles, the triglycerides and the VLDL were primarily linked with the alpha diversity. The presence of higher neutrophils and white blood count, even within what is considered within normal limits, also negatively correlated with health indexes. And what was interesting also was that people who had family history of ischemic diseases in their family practices a better, probably, this is in interpretation, probably a better lifestyle that correlated with better indexes. So to conclude, we had this nice cohort of large and deeply characterized across different dimensions of health. And we think that the gut microbiome is linked with various aspects of human health, including lifestyle, diet, diseases, medications, and overall well-being that each subject reported. This suggests that it may serve as a complementary lab work that we can do on routine checkups. Like future direction or future question that we are trying to answer using this cohorts is, can longitudinal changes, either in the different lifestyle or different disease status, can direct the personal changes to a good or bad condition, and can we use this to advise each person if they did the right or wrong change for their microbiome or their general health? And the more interesting question is, how much can this predict the health outcome? And we hypothesize that it may predict health outcome because just from this couple of months, recent months, there were a lot of publication in the Nature Medicine, for example, showing that in order to achieve a healthy aging, the key is really this lifestyle changes, including diet and different physical activities. And also this interesting paper from last month showing that introducing a bad diet to the African heritage really affected both the metabolic, the microbiome, and the immune relation to this bad diet. again implicating how important this to our health in general. So I want to conclude and thank the different people that were involved in this project, the students in the lab and the people who gave us the samples. And these are the current and past funding agents. Thank you.
Thank you. We have time for just one very quick question. Do you want to? So I think your findings are interesting, and it's consistent with what's been shown with the Healthy Eating Index based on the last dietary guidelines for Americans, where it's higher in younger ages, it dips down in like young adult, and then goes back up. So it's definitely consistent. So what do you think accounts for improved lifestyle? Is it more just concern of health given the increase in health problems as we get older?
It may be that, but also I think our habits deteriorate as like the years go by. So someone who is 80 now has incorporated a healthier environmental exposure when he was 3 than someone that is 50, and so this I think is another component in addition to what you're suggesting, I think.
Great, thank you so much. Very nice talk.
All right, terrific. Our next speaker is Jennifer Labus from UCLA. Their talk will be entitled Metabolically Derived Dietary Intake, Links to Gut Microbiome Composition, Gut Hormones, Cognitive Markers, and Brain Imaging-Derived Phenotypes in Healthy Participants.
Thank you for staying. Good afternoon. Diet has been associated with health and disease, including cognitive functioning and mental health, and impacts brain structure and function. Poor diet has been linked to brain atrophy in sensory motor and executive control regions. The human diet impacts the composition and function of the intestinal microbiome, influencing health outcomes. There's also a well-recognized lack of reliability in self-reported dietary intake due to recall and social desirability biases. Therefore, there's great interest in objective dietary markers assessed with metabolomics that might help bridge the gap between self-reported dietary data and actual food intake. So the aim of this study was to link a metabolically derived dietary intake pattern or profile with gut microbial community composition, gut microbiome-linked metabolites, gut hormones, cognitive markers, and brain imaging-derived phenotypes in healthy participants. So our sample consisted of 111 individuals, 80 females, with a mean age of 33, ranging from 18 to 60, with an average BMI of 31.9, so they would be considered in the obese range. But we did have a range of subjects between 19 and 51 BMI. These subjects underwent clinical phenotyping, neuroimaging. They also provided fecal samples for 16SR RNA sequencing, as well as metabolomics, which we did both through Metabolon's platform to get feature tables for gut microbially linked metabolites, as well as another platform from UCSD that Peter Dornstein talked about today that essentially is for bile acids and several newly discovered bile acids and their conjugates. And a subset of these individuals also went through gut hormone profiling. So in this study, the dietary profiles were empirically derived from untargeted metabolomics data using a novel metadata-based source annotation method developed in the Dornstein lab at UCSD here in San Diego, and it's referred to as foodomics. The picture on the left depicts the process of MS2 spectra matching to food mass, which is a database of food and beverages used to determine food and beverage consumption based on the mass spectra data. So based on the HITS feature tables containing counts are generated at different levels of ontologies, and then we can derive dietary profiles through principal components analyses. On the right, we can see the results from matching the MS2 spectra from cyanidin, which is a plant pigment responsible for the reddish purple color in fruits and vegetables. And as you can see, the search returned 47% prevalence for teas and fruits at the level 4 ontology. So over here, we can see the teas having the most hits. And then you can also see that the highest prevalence then at level 7 ontologies we're seeing in blackberries, raspberries, and strawberries, as we would expect, sort of trying to give you some face validity for this platform without complicating its explanation. So we did 3T structural MRI scans in these individuals. We computed the gray matter volumes for regions of interest in the executive control and sensory motor networks, where poor diet has been linked to brain atrophy as well as cognitive decline. And then performed general linear models controlling for age, sex, BMI, and total intracranial volume for brain imaging to look at the associations between this empirically derived dietary profile with the clinical brain and gut microbiome assessments. So here you see that we performed by principal components analysis on the centered log ratio transformed raw food counts at the spectral level. And then we can derive a dietary profile looking at nonlinear associations between the principal components and the relative abundances at the level 3 ontology. And so what we can see is that high scores on this dietary pattern reflect an increase in meat, dairy intake, and a reduced intake of fruits and vegetables. And it was very normally distributed in this population. This dietary profile was linked to several gut microbial genera. For example, the diet was linked with the abundance of several species and genera belonging to the Clostridia class, which play a key role in bile acid production. In addition, I don't have time to go through all of these, but several of these genera have been linked to diabetes, obesity, inflammation, cognitive decline, and Alzheimer's disease. And all these were significantly different at FTR correction of 0.05, so pretty strong results. Oops, I just ruined my, okay, sorry. So next we looked at microbial linked metabolites within the Metabolon platform, and we saw that the diet was associated with several of these metabolites, and which have also been linked to inflammation, neurodegeneration, cognitive decline, and insulin production. For example, imidazole, propionate, and TMAO. They're both produced by the gut microbiota and are linked to impairment in insulin signaling, type 2 diabetes, and have been implicated in the pathogenesis of Alzheimer's disease. We also looked at these newly discovered as well as some annotated bile acids and saw that poor diet, so greater intake of meats and less intakes of fruit and vegetables were associated with reductions in these bile acids. And so in particular, we saw many microbially generated bile acids, deoxycholate, lithocholic acid, amino acid conjugated bile acids, including threonine, glycine, tyrosine, and glutamate conjugated bile acids. So then we looked in a subset of individuals that had gut hormone profiles and also saw that higher scores or poor diet was associated with increase in C-peptide, GIP, as well as GLP-1. And just want to point out in terms of interpretation, all of these are linked to, involved in the regulation of glucose metabolism, insulin secretion, and appetite, and have been linked to obesity. So these findings suggest that individuals with poor dietary patterns may exhibit early physiological signs of insulin resistance, as indicated by the elevated C-peptide and GLP-1 levels. The increase in GLP response could represent a compensatory mechanism to maintain glycemic control in the context of excessive caloric intake or adiposity. Together, the hormone patterns are consistent with dietary-driven metabolic stress that often precedes obesity-related complications. We also saw links with clinical phenotypes. So we saw that a less healthy diet was associated with greater reports of self-reported language difficulties. So we did not give people like executive functioning measures like you would in AD, but the MASC is a measure that kind of measures subtle cognitive difficulties in healthy populations. So we saw difficulties in this poor diet was associated with difficulties in language. So this, one of the questions is, when talking, I find difficulty conveying precisely what I mean. We also saw that poor diet was associated with lower mindfulness scores, which can be considered a proxy or an index of attention and cognitive flexibility. And sort of as a manipulation check, although not saying they're the same, I used that alternative healthy eating index and did see that poorer diet was negatively correlated with a higher alternative healthy eating index. But again, eventually, hopefully, we can detect these dietary profiles empirically, and we will not have to rely on sort of self-report. And not pictured here, the poorer diet was associated with increased perceived stress in individuals as well as lower resilience. We also looked at the brain and saw poorer diet was associated with decreases in the volume of executive control regions. We know that this is atrophy in these regions is sort of a prodromal sign for Alzheimer's disease as well. And we also saw atrophy or decreases in the volume of sensory motor regions that was linked to poor diet in this population. So in conclusion, higher consumption of meat along with reduced consumptions of fruits and vegetables was associated with reduced gray matter and cognitive control in sensory motor regions, as well as self-reported neurocognitive difficulties in the language domain and lower mindfulness, reflecting less attention or lower attention and less cognitive flexibility. This empirically derived poorer diet intake was associated with a less healthy gut microbiome and generally linked to diabetes, obesity, cognitive decline, and Alzheimer's, as well as cognitive vascular disease. And these individuals with poorer dietary patterns may exhibit early physiological signs of insulin resistance, as indicated by the C-peptide and GLP levels. And in some of these findings provide further evidence that dietary intake may affect cognitive process through the brain-gut microbiome system. And so that I just want to thank my co-authors and collaborators, especially the MS collaboration with UCSD, Peter Dornstein and Ipsita Mohanty, Jasmine Zellman, as well as my coworkers at UCLA. And senior author, Dr. Gupta, or Dr. Church, my fault. So thank you. I appreciate you coming.
This presentation's open for questions.
Thank you for a great talk. I think your reference-driven data annotation system is incredibly powerful, especially with the limitations of the recall bias on those food frequency questionnaires. So my question is, what kind of validation has been done on backmatching, what you're seeing in the diet from this reference spectrum analysis to what the participants are actually consuming?
So, right. So this is what we refer to as Foodomics 1.0. There's already another version, and they're they're working on that. Because I'm not completely clear on the specificity, we're not using level 7. We're sort of saying it's in this category of meats or food or fruits. Because you could be had a strawberry or you could have had strawberry tea, right? And so I think this is a novel approach that they're taking. It's incredibly strong, and they've already cataloged over like 4,000 different food items, and they're just working on improving that now and looking at the validation. But many of these came from they gave people a specific diet or an apple, and then they find out what the spectra are and try to match it. But yeah, I mean, in terms of specificity and validation, it's a work in progress that these guys are just really good at what they do. And Peter Dornstein would be the one to tell you more about the technology involved, but feel like it's a very good platform for thinking about this instead of relying on these self-report measures.
Yeah, and since you're using the MS2, do you find that certain features are less detected? Especially when they go through degradation?
I think, I will say, for example, the bile acids platform. So those are more detectable in fecal samples versus serum. So the sample matters. Fasting status matters, things like that. I'm not an expert in MS2 by any means, so I can't really probably answer that to
problem.
your...
Thank you.
Okay.
Time for one more question.
Hi, thank you for the talk. I just had a question on foodomics. How stable are they? Are the foodomic derived dietary profiles over time? And can you use can the temporal changes correlate to the longitudinal changes in cognition and brain function?
Yeah, so there's been work with foodomics looking at the mind diet over time. I didn't include those references, but if you look up the Dornstein lab, they have looked at changes over time and linked those to improvements in executive functioning in healthy individuals and individuals with AD. So it's quite a powerful method longitudinally, but again, this is really pretty brand new, just now seeing papers published. So if you look up the senior author Dillmore, there'll be a couple of nice studies that looked at foodomics and changes over time.
Thanks very much.
Okay, thank you.
All right, next up we have Weijia Wen from Sun Yat-sen University, and the title of this work is Dietary Whey Protein Regulates the Gut Phagome Bacterium Transkingdom Interaction Dynamics to Attenuate Intestinal Inflammation and Crohn's Disease.
Thank you, because we didn't get his visa approved, so I have to represent him to present our study. So my name is Tao Zuo from Sun Yat-sen University, China. So the title of my talk is Dietary Whey Protein Protects Against Crohn's Disease by Orchestrating Cross-Kingdom Interaction Between the Gut Phagome and the Bacterium. So our research focused on Crohn's disease. Crohn's disease is a subtype of inflammatory bowel disease, and the pathogenesis of CD is associated with a changing diet and lifestyle during urbanization. And urbanization and diet and lifestyle can all change the gut microbiome, including the bacterium and the virus and phagome. So our lab's focus has been on the bacterium as well as the virus. Actually, virus is our main focus because we think the virus is actually the quantity is 10 times larger than the bacteria. So we want to focus on the viruses, and most of the viruses in the gut phages. So one question is what specific diet and how it affects CD and biomodulate the gut microbiome, including bacterium and the phagome. So in our previous study, we have been focused on the phageme. We found that phageme actually, dysbiosis is more robustly associated with inflammation when comparing to the bacterium. And the phage dysbiosis predicts bacterium dysbiosis. And when we isolated the phageme from the CD patients and give that to animals, and it can elicit a stronger inflammation phenotype. So what remains unknown is what are the protective phages that may interact with bacteria to counteract inflammation. So regarding phage, we know that phage can prey on bacteria and in a lytic cycle or lysogenic cycle. And there are studies, anecdotal studies showing that simple sugars which are contained in diet can induce phage release. But overall, it is unknown how diet can regulate the phage bacteria dynamics in the gut and how that relate to the CD pathogenesis or protection. So we conducted this study with the overall goal to find a specific diet that may protect against the CD and the underlying mechanism in terms of regulating the gut phage bacterium interaction. So here is our study flow. We firstly screened a diet and want to find a diet that might regulate the phageme and the bacterium. And we found a dietary component, which is whey protein, can play an anti-inflammatory role in vivo. And then we want to ask whether whey protein can causally reshape the phageme and the bacterium. And then we want to ask whether the reshaped phageme and the bacterium can actually causally counter inflammation. And then we identified the key phages and bacteria that mediating the anti-inflammatory phenotype. And we'll then look at the mechanism and we'll validate that in multiple animal models. And overall, in part one, we started from humans by including healthy controls and patients with Crohn's disease, and did dietary questionnaire investigation, and then we did phageme profiling and bacteria profiling respectively. While when screening the dietary effect on gut phageme and the bacterium, the first and foremost come to us is a whey protein, and it influenced the phageme strongest, while it also influenced the bacterium, but the effect size on the phageme was larger than the effects on bacterium. Then we looked at the whey protein's effect on IBD, its association with IBD. We went to the UK Biobank database to look at the association, and we found that whey protein intake was actually associated with a lower risk of CD, which indicates that it's a protective factor. And then we go back to the whey protein's effect on the gut phageme, the bacterium. We found that actually whey protein consumers has a phage bacterium ecological network that was more similar to healthy individuals than compared to its similarity to CD patients. Then we reasoned that whey protein may protect against the CD through modulating phageme rather than on bacterium. Then we moved to animals to just validate our hypothesis and to test whether whey protein play an anti-inflammatory role in DSS mice, and the result is yes, whey protein can attenuate intestinal inflammation in animals in DSS mice in terms of both disease activity index, endoscopic assessment, colon length, and so on and so forth. And then we move to part 3. We want to look at whether whey protein can actually reshape the gut bacterium and phageme. Firstly, we look at the phageme in mice. Again, so whey protein can actually configure the gut phageme and increase the phages that can target on the pathogenic bacteria, including fern viruses and other viruses. And at the bacterial level, we also look at how whey protein can change the bacteria. Actually, whey protein increased the bacteria, including Streptococcus thermophilus, which is a probiotic bacterium, while it decreased the bacteria, which is a pop star, Commencia muciniphila. We call it AKK. So Considering that whey protein can shape both the gut phageme and the bacterium and counters intestinal inflammation, so we want to ask whether the reshaped phageme and or bacterium can causally counter the inflammation. If so, which one is more important in terms of phageme and bacterium? So we then conducted a part 4 study to compare and elucidate the counter-inflammation effect of phageme versus bacterium reshaped by whey protein. Basically, here is what we do. We give mice whey protein, and after whey protein administration, we isolated this phageme and phageme and bacterium and gave that to the recipient mice, respectively, both of the bacterium and phageme. And the result turns out to be the phageme, the reshaped phageme by whey protein is most influential in terms of ameliorating the intestinal inflammation, which is very interesting. Then we speculated that phage may play an upstream role in terms of counteracting intestinal inflammation. It may regulate the bacteria downstream and then relate to the host inflammation amelioration. Then we want to ask which phage and which bacteria are the main player. So we conducted part 5 experiment to identify the key phages bacteria that may play the role. And basically, we overlapped our findings in humans and in mice. Mostly, we want to start with the bacteria because the bacteria might be the ultimate player. So we overlapped the bacteria that were enriched in humans and in mice that were administered a whey protein, and we found Streptococcus thermophilus, which we call it STH, was actually enriched. But when we give the whey protein shaped phageme to infect the STH, the result turned out to be negative. So we speculated that maybe there is an intermediate bacteria mediating the phage's effect on downstream to the STH. And then we want to find the intermediate bacteria, which finally we found AKK, because AKK was decreased by whey protein, but it negatively correlated with STH, and meanwhile it negatively correlated with AKK phage in the gut. So AKK might be a downstream bacteria. So starting from there, we isolated the host bacteria AKK and its phage in-house, and then cultured that with the 3 members, the AKK phage, AKK, and STH. Look at the abundance, and it just validated our hypothesis that actually the AKK phage can inhibit AKK, and downstream it unleashes the growth of STH. So we then added the whey protein in the system to look at whether whey protein can promote phage release from AKK, and the answer is yes. Whey protein promoted phage release from the system and decreased the abundance of AKK. So it comes to part 6 because we want to test that in vivo to validate our hypothesis of especially the counter-inflammation effect of AKK phage, AKK STH axis. So basically, we gave the mice STH, AKK, and without AKK phage. Then you can see that if you give the AKK phage to the tripartite members animals, then the intestinal inflammation was more ameliorated. But if without the phage, then the mice showed a stronger inflammation phenotype. And then lastly, we want to validate our finding in another animal model just to validate the counter-inflammation axis of the phage bacterium interaction. We did that in a TMBS model. Here is what we do. We give whey protein. First, we gave them AKK phage, AKK STH. Then we gave them without whey protein, and it just again validated our hypothesis. If there is whey protein, then it can protect the intestinal inflammation. And one thing I want to stress is that when we look at the mucus layer by immunofluorescent staining of the mucin 2, then we notice that if there is no whey protein, then AKK can degrade the mucus layer because AKK is a mucin encroaching bacterium. Because if you don't have whey protein, the phage cannot be released. Then the AKK and eat the mucin. And finally, this is our graphical summary, which I can reiterate that whey protein can promote phage release from AKK. Then AKK the abundance of AKK can be downregulated while AKK phage abundance increased. But AKK can inhibit STH. STH are probiotic bacteria. If you have whey protein, then the AKK's abundance is decreased. So STH's abundance can be increased. So STH can protect the mucus, while AKK's abundance decrease cannot degrade the mucus layer. So with that, I want to conclude my talk. I want to thank my lab members, my PhD student Renping, and my postdoc Wei Jie, and thank you.
Excellent talk. We have time for a couple questions.
I can start. That was great. One thing that strikes me is strep thermophilus is also often found in dairy products.
Right.
Is the strep that you're seeing in the gut from the whey protein, or is it inherent to the mouse gut?
It is inherent in the, in mouse gut, they don't have really too much Streptococcus thermophilus because it is usually administered through fermented milk. But for whey protein, they don't have STH. Because whey protein is just whey protein. If you give them fermented milk, yogurt, then it has STH.
I have a question as well. In the initial transplant model where you introduced the bacteriome or the phagome, the recipient mice, were they on the whey protein
No,
diet?
they were not. So we want to just eliminate the dietary effect. We just want to look at the microbial effect. Therefore, we just filtered the stool and gave that to the recipient mice.
So what do you think you would have seen if you did, plus or minus the whey protein diet with the recipients?
So basically, we just transplanted the bacterium and the phagium to the recipient mice, and we just found that the phagium that were derived from the whey protein administered mice caused the more attenuated inflammation phenotype.
Okay, yeah, I was just curious if maybe you would have seen like more equivalent effects with the bacteriome.
No,
You know what I
the
mean? But-
phagium are more, the phenotype was better.
Got it. Thank you.
Thank you for the interesting talk. I'm Catherine, dietitian from New Zealand. I had a question when you were looking in the UK Biobank and you looked at whey protein. Was whey protein, was that just a marker of dairy intake, or was it specifically whey protein?
I think it is specifically whey protein.
All right, thank you so much.
All right. Our next speaker is Xuan Yang from Imperial College London, who will be telling us about impact of dietary heme on circulating, hepatic, and gut microbial metabolism in germ-free mice colonized with a defined microbial community.
Okay, so good afternoon, everyone. I'm Xuan, and I'm a currently PhD student in Imperial College London. So today's my talk is about impact of dietary heme of circulating hepatic and gut microbial metabolism in germ-free mice model colonized with a defined microbial community. And I'm really honored to present my current project as an early stage researcher at DDW. So, well, I will just start with the background of heme. Actually, heme is a mantle porphyrin, which contains porphyrin and one iron cation, and primarily found in animal sources like red meat. And heme is primarily taken up by the duodenal and proximal jejunum of the small intestine, and can be transported within and between cells. So here actually is a structure of heme. You can see in the middle is iron, cadmium, and the structure is porphyrin. And then I will introduce some benefits about and as well as a risk about heme. So actually, heme plays a crucial role in various cellular function and overall health. On the one hand, it enhanced iron absorption and is very, very crucial for oxygen transport and storage. And it always can reduce the risk of iron deficiency. But on the other hand, it always involved in anemia, inflammation, as well as related to the increase in CRC risk. So here is a simple example. We can see if the higher red meat intake, it always linked to the higher CRC risk. And there is a growing evidence shows that heme metabolism linked to the CRC risk. It may involve DNA damage, ferroptosis, inflammation, as well as changes in the gut microbial composition. And this is what our work steps in. About this pathway, the gut microbiota plays a key role, but how the bacteria and host metabolism affected and what do they produce, does this metabolites link to CRC, is the key question we're exploring. So we use the gene-free mice model with a defined microbial community. This allows us isolate the heme impact without interference from the unknown or complex or uncontrolled models. So we choose the bacteria is always involved or promotes the colorectal cancer. So here is our objective. So we aim to investigate the impact of heme supplementation on the circulating hepatic and gut microbial metabolism in gene-free mice model colonized with a defined bacterial community associated with CRC risk. And this is our method. So I hope this figure is easy to understand. So we just give our bacteria using our gavage, and then we introduce our heme diet in the week 14, and at week 18 we just do the counting. So the gut, liver, plasma, and urine samples were collected, and we use the NMR to do the sample analysis. So the PCA and OPLSD models were used to identify the key patterns and biomarkers in our experiment. So now I will show you the results. So you can see at firstly, the second content, urine sample, liver sample, and plasma samples. In the both, on the left side is PCA models and OPLSD models. They are both overlap. So that means there is no significant difference among these samples. But as we move on to the colon content samples, things become interesting. So we can see in the PCA models as well as in the OPLSD models, there is a significant difference between the heme group, blue, and the control group, which shows in the red. That means the dietary heme induced a distinct and statistically significant metabolic changes. We always exclude the effect of sex, so we just use a PCA models. So it revealed there is no significant difference among the female and the male in the colon content samples. So to identify the key metabolites in our experiment, we look at the OPLS loading. So you can see the standout peak pointed out it potentially is methionine. So to confirm this metabolites, we just use the spiking test as shown on the right-hand side figure. So you can see when we add the pure methionine in one colon content samples, the peak is overlap in the same retention time. So that means it's methionine. So based on these results, there is an interesting part because we're curious the chyo amino acid, we know that there is a different form of amino acid, L amino acid and D amino acid, and they always show some different biological function. So we use the same colon content samples to figure out how about the L-methionine or D-methionine in my samples. And also, I really want to know if there are any changes cross into the circulation. So that's why we choose the plasma sample as well. So in this stage, we use LCMS to analyze. So here is the result. As you can see, I use the same color before, like the blue represents the heme group and the yellow red represents the control groups. So both in the first 4 figures, there are L-methionine and D-methionine in clone content sample and plasma samples. They always show significant higher in heme group. And more interesting thing that I always compare like D and L ratio here. That means you can see the D-methionine showing more pronounced increase in heme group. After observing this result, I really want to know the correlation between these 2 samples. So I did the correlation. So you can see in the D-methionine or L-methionine, it always show, as showing the p-value, so it always show the positive correlation among the samples. So that means maybe it's potential link to the diet, host gut microbiota, and the systematic exposure. So here is our key findings and discussion. So our finding is heme supplementation significantly increased both colonic luminal and circulating methionine levels, especially the D-methionine, and it suggests heme influenced the bacterial metabolism of D-methionine with a defined bacterial community linked to the CRC risk. And also, I always did some research about the methionine and the CRC risk. So you can see actually so far the publication is still controversial. For example, they mentioned high methionine may be negatively correlated with CRC risk, but they always there is always mentioned that methionine maybe can reduce inflammation. Also, the D-methionine may protect against the CRC, but it's always need more research on their specific roles and effects. Also, I'm really curious about heme methionine and CRC risk, this relationship, but there is a limited research significantly examining these combined effects. So based on this, I really here is our future direction. So firstly, we want to identify what bacteria species with this defined bacteria model produce methionine in response to heme supplementation. So for example, we want to use a single species bacteria culture. And then we want to investigate the impact of heme in a complex microbial community to explore the potential role of methionine heme in modulating colorectal cancer risk. And also based on this result, maybe we just move on to the cancer model to figure out more findings. Yeah, at the end, I really want to thank my colleagues and collaborator, as well as my supervisor, Professor Julian Mackenzie and Dr. Jia Li. And also, if you want to interested in this talk or want to contact in the future, at the right-hand corner here is our contact information. And many thanks for your attending today. I know it's a last session of this conference, so many thanks for listening.
Presentation's open for questions.
Thanks, really interesting talk. So I have a question about your heme supplementation in the germ-free mice. So was this given as just a one-shot oral gavage, so all the mice got the same, or was it an injection?
Actually, we just made the food pellet. We just use the heme into the pellet to give the mice. So the oral gavage we just use to give them bacteria.
Mm-hmm.
Yeah.
And so were you able to confirm that all of the mice consume the same amount of heme food?
Sorry?
Were you able to confirm that the mice all consumed the same amount of heme food?
Actually, it's a good question. We cannot confirm during our experiment because we just collect the gut content, whole gut content. We just confirm that the heme is actually absorbed in a small intestine. So about the colon part is no heme. So yeah, that's a key question we will explore.
And just one more question. Did you try to supplement germ-free mice without your defined consortium with the heme to see what proportion of these metabolites and the methionine were due to de novo synthesis by the mouse instead of the microbiome?
Yeah, so far we didn't, but I think, yeah, it's a good question because we always concern if it's a direct link, like heme direct link to the methionine, or just indirect link. So we always concerned about that. So that's why we want to use more complex model in the future to see this mechanism. Yeah, thank you.
Hi, very good presentation.
Thank you.
So previously in your study, you inoculate the Klebsiella and also other bacteria. So I'm wondering, like, what kind of Klebsiella bacteria that you inoculate?
I just can't give you the strains because this Klebsiella we just isolate from the bariatric surgery patients. So we do the sequencing for this species, but it's hard to say which the strains of this Klebsiella.
Okay.
Yeah.
What about the species?
Yeah, I think I have give the name. Maybe just a second.
Yeah, I saw it's Klebsiella.
Oh yeah, here. Yeah, this one. This is what you want?
Oh, it's mission. Okay. And also in your study, you find the higher concentration of the methionine.
Yeah.
You use that is on target or target. I'm not sure because
it's on target.
It's on target,
yeah,
right?
Yeah.
So maybe it will also affect the one-carbon metabolisms, like methionine cycle, something like the metabolism of the methionines. Do you have any insight on that?
So far, not.
Okay.
Yeah.
And also, I think like methionine is also related to the antioxidants like theanine, so maybe you can look at it.
Okay.
All right.
Okay. Thank you. Thank you so much.
All right, thank you.
All right, last up we have Robert Gans. The title of this presentation is Open Label Study of a Novel Fiber Product for Bloating, Gas, and Bowel Regularity.